so slow, so easy, or is it okay? Okay, so we were in the Hodgkin-Huxley model. So we know more or less the ingredients that are important to reproduce an actual potential. We can measure that in the neuron, and we can do the propose the equation. So now we have two options. We can go to put more biological ingredients, or we can go into simplified models. So we will start going to more biological ingredients. Okay. So in the usual Hodgkin-Huxley model, we still have this capacitance times the derivative of potential in time equal to the sum of all the currents. And then I try to convince you that with the sodium and potassium, just here is, it could be just a IK here, and a leak current, that it's everything else, we are able to reproduce in a very good way the neuron that they measure. But people start to measure different neurons from different animals, from different areas, looking for calcium, looking for different behaviors of potassium, and there is all possibilities. So if you are really interested in specific brain region, you can add here, and different papers will add and call modified Hodgkin-Huxley models. And some of them are more important, for example, for plasticity, and some of them are important for response, like adaptation or bursting. So, because so far what we look is just the actual potential of the neuron, but we're not looking what else is going on. So I described here the actual potential. I said, okay, I had the neuron here. We did any kind of stimulation, and then pass a threshold, and then there is the actual potential. And this is time, and this is the potential. But now, if I repeat the stimulus, or if I put a constant current, what's going on with these neurons? So we can change the scale, and the neuron are going to do something like that. So we have, sorry, can't see. We have a behavior. So will be this neuron periodic? Will be this neuron stochastic, like these fires, how it's going on for different external stimulus? So depending on the kind of question, you add different currents that are important to have different behaviors. And electrophysiologically, we are going to see in a while, but we can have many different behaviors. So neurons that are spiking periodically, neurons that are bursting like trrr, trrr, trrr. So we are not interested just in reproduce the actual potential with these 4 differential equations, but we can also be interested in reproduce other electrophysiological behaviors. So we can add different currents that help us to do that. Specifically, if we look to some kind of neurons, if we look to the firing frequency for the external current that we applied, if we applied a current that is small, some threshold, nothing happens, so there is no activity, so there is no firing rate, or just one actual potential happens and nothing happens then. But then if we go to a specific threshold, we can have discontinuity or continuity, but then the neuron starts to fire with a specific firing rate. So if we are stimulating here with a current or with a higher current, the behavior of the neuron, a higher current will be different. So it will be fast. So we can explore this external input to see what's going on to the neuron, and sometimes this is important for these behaviors, different currents that we are at here, but we can go. So the other way of going to more detailed things is consider detailed compartmental models. So now we consider not the neuron as a point and 4 differential equations for this part, but we would say that we have many compartments here, and we have a branch of differential equations for this part, and then again for this part. So the same 4 differential equations of the Hodgkin-Huxley neuron, but perhaps different parameters along the neuron. So this is a cable equation. So we can now use the spatial relation, not just temporal, so dv dt, but we can also have something like dv dx. So along the space, and then we can complicate a lot of our life, like we can say the cable model, and then there's dendrites and axons, they can ramify, so we can have now one circuit, like for the beginning, we start with just one electrical circuit, would represent one neuron. But now the neuron can be represented along the space for many different electrical circuits. So these compartmental models can be useful for specific things, like if you would like to know how this ramification is important for the information, probably it's different, this neuron and the neuron with a small tree of dendrites here, so the common neuron there. So we can complicate it as much as we can and depend on the type of question that we are interested. So if we want pharmacological manipulations, should we put here in the beginning of the soma, or should we put here more in the end of the neuron? Or there is a question like, is the form versus the functions. So how different will be the behavior of the neuron if it has a different shape? So then we can complicate or go into more details. So if we want to compare our results, like the external current that we put in an experiment that we just clamp, really one neuron in different regions, and we want to see the difference of this activity if we deliver a drug here or a drug here, then could be important to really model all these details, how this was branched in different directions, so how this signal propagate here. But then if you want to compare our model and do a simulation of hundreds of neurons, probably it's not necessary to go all to this detail, and it's enough to use one compartmental model, or sometimes just 2, to make sure that there is some difference from the soma to the rest of the neuron. So if you want to compare to experiments that are measuring really one neuron, now you can have this inside the brain, or in a tissue, or in slice. Then could be more important to put all the details, to put, there is a difference in the diameter of the axon, so the conductance can change with this diameter. So there are a lot of details, and there are people that really works in this scale. So they are not interested in put many neurons together, but they are interested in understand what kind of difference in small parts of the neuron change the behavior of the neuron. So typically, this is not so useful when you are trying to do biological neural networks, because in these networks, perhaps this is not so important in the collective behavior. Probably someone will find specific cases in which, so if nature is like that, it's probably important for the collective behavior. But typically, for computational reasons also, we just go for these more detailed compartments if the kind of question is related to this function that really need to see in one neuron activity. When we want to look to a collective activity, it's easier because you have to do a lot of assumptions in the model. When you add more variables, more compartments, you have to make sure the conductance of sodium in different compartments. So you have to choose a lot of things that sometimes it's not helping you, like to have a better biological neural network, but so many ingredients, okay? But it's always a compromise. Any question? So we can have these modified Hodgkin-Huxley models, which means that we can add different ions, or we can add this information of space. Typically, this is the two main differences that we can go in order to do more biological models, modified Hodgkin-Huxley models that have even more biological details. To the other side, we can go now to simplified models. So, okay, we have this Hodgkin-Huxley model. It works well with 4 differential equations. But if I want to do a simulation of lots of neurons, perhaps I don't need all the 4 equations or all the details about sodium and potassium, and I can have just electrophysiological plausible values. In order to do that, we can go to these simplified models. This is typically what we do when we are doing biological neuronal models. So we go to simplified to put more neurons together. But, I mean, you could do networks of many of more biological neurons and see what's going on depending on your question. So the first, no, not the first, I mean, if we start from the Hodgkin-Huxley, typically we can talk about reduce the number of differential equations. So we start with 4. How can we go to less or 2 or I don't know? So the easy cavity model is a very good compromise between simplified going from 4 equations to 2, but remain, I think it's here is better. So, but remain with this biological plausibility in the sense that the model can reproduce many firing patterns, okay? So the neuron doing an axial potential, but the neuron do sometimes periodically, sometimes in bursts or shattering, like trr, trr, trr. And then easy cavity did a model that we cannot recognize anymore sodium or potassium here, but we have a differential equation for the potential and a recover variable, but in this system, we cannot say, oh, this is more sodium, this is conductance of potassium, but we have 4 parameters here, A, B, and in the reset condition that I will talk in a while, that changing these parameters, A, B, C, and D, we can change the behavior of the neuron when you put a specific external current. Does it make sense? So now we lost the information of real biological details about sodium and potassium, but we can have, changing the parameters of the model, we can reproduce the behavior that we really measure when we measure brains in slices or brains in vivo. These differential equations, you need 2, but you need reset conditions. That means that when it reaches the threshold, when V is 30 millivolts, we say that this is reset, and then we change the V by hand. We say, okay, a spike happened, now come back to the beginning and start again. In the Hodgkin-Huxley model, we didn't need this reset condition by hand. So can you see there is gains or... but then we can have this neuronal variability, and we can also I will talk more in synapses, but we can also simulate excitatory and inhibitory neurons. So excitatory neurons have different behaviors from inhibitory neurons, which are neurons that send inhibitory synapses. I will talk in a while, but they can be these fast spike and low threshold spikes. If you put the same current in these neurons, just because they have different parameters of the model, they would behave differently, okay? And typically, this behavior is for inhibitory neurons, and this behavior is for excitatory neurons, so the same model can reproduce different neurons from different areas. And also here is a thalamocortical neuron, so these neurons are typical from the cortex, but these neurons are common in the thalamus. So all these details were not possible with the Hodgkin-Huxley model. The Hodgkin-Huxley model, unless you add the modified currents and put slow calcium or slow potassium, you would have always the same behavior, that the firing frequency depends on the external current. And here we can see that not just the firing frequency is changing, but also the way, if it's periodic or the pattern of the spikes. So firing spikes are very important, and there is an ingredient. I mean, if you take a tissue, if you take a piece of cortex, you're going to find a proportion that is more or less known of regular spikes neuron, intrinsically burst neurons, and shattered neurons. So if you like to reproduce a part of the cortex, it's better to have neurons that are doing that or that or that, just changing these parameters, instead of simulate your neurons with Hodgkin-Huxley and really try to determine different sodium and potassium for them to make different patterns. Okay? It's not necessary. So another way of simplify even more, and then we can use the leak, integrate, and fire model, is going for just one differential equation here that look like a capacitor that you're just charging, and then you say, if I charge a lot, I will arrive in the threshold, and if I arrive in the threshold, this is a spike. So this is a very simple model that if you put an external current here, that is not enough to make the potential arrive to the threshold, nothing happens. So you put an external current constant here with this small value, and the neuron do not spike. So just charge, I mean, stay here. But then if you put an external current that is fixed, but that is sufficient to make this increase go until the threshold, after the threshold, you say by hand that a spike happened, and then you have this reset condition, and then you start again. So if you put an external current here, this leak, integrate, and fire neuron spikes periodically, okay? But if you not put a constant current, if you imagine that the external input is stochastic, it's arriving different currents here, so you will have a spike pattern, a spike train that look like random because of your external noise is random, and sometimes you achieve the threshold, sometimes not, okay? So sometimes you spend a little bit more to achieve the threshold. So for noisy external stimuli, these neurons can fire not periodically, but because of the external stimuli, they would have different spike time intervals. And for those who like dynamical systems, here it's quite simple to see what's going on with these neurons. If we have this differential equation here, dv dt as a function of v plus re, we can do change time here, and we can look for fixed points. So if we plot v point, dv dt here, and v here, we can find as a fixed point here the I, and which means that we have this line. So dv dt is minus v plus, it's the equation of this line here. So y of v is y of x is a of x. My God, in English it's hard. So it's an equation of the line. So it's just that. It's just v point as a function of v is just the line. So it means for those who like dynamical systems that if you are here in this region, you are going more to this point, and we are going more to this point. So you have a fixed point here in y. And if y is larger than 1, you go to a different place here, and we are not going to the fixed point. So depending on your threshold, you can go to the fixed point and just stay there and just stay here. And if your fixed point is there, you pass the threshold, so you would go further, and then spikes happen. So I think, so the neuron keeps firing regularly above the threshold. So this is this case here. It's okay if you are not from this mathematical background to look this phase space, don't worry. Just if you want to do calculations, this leak integrate and fire is better than calculations by hand is better than a Hodgkin-Huxley model with 4 differential equations. So people sometimes want to reduce the model because you can do calculations by hand. You can find more the feeling of the math, what's going on with more rigor. And sometimes just because it's easier to implement for computational reasons. So it's, again, a choice for compromise. So finally, there are other simple models, Fitzhugh-Nagumo. You can analyze nucleons in the phase space or Morris-Lecard. There are a lot of different types. I don't want to go in all the details of the different models, depending on what you want, but these spiking versus firing rate models are very important to our comparison. So for artificial neural networks, we are using neurons that are more like a firing rate model. So these neurons in our perceptron, if we have inputs arriving in the neuron and it is doing something like a sequence of spikes. Look here, I could say that it's 0, 0, 0, 1, 0, 0, 1, 0, 0, 0, 1. So the state of the neuron, I forgot about all the shape of the actual potential, and I'm just looking if there is a spike. But I still have time here. So a spike happened here, many spikes happen in this moment. So still I have spikes, but I can go to a different kind of neuron in which I don't look to the spikes anymore and just to the firing rate. So here the firing rate is higher than here. We have more spikes per second or per time, whatever. And these neurons looks like much more with the neurons that we are using in the artificial neural networks. So they are the default neurons of AN, and we look for this average of activity. And then we could write the state of the neuron. This is what we saw in the previous course. It could be that the state of the neuron is the sum of all the weights that come before, the inputs, and then there is this transfer function. But in the end, we're going to look to the activity of this neuron. Okay. So in neuroscience, like a real for the brain, there is a question, what is important? Is it important firing rate models or spiking time? I mean, not just to simplify and do artificial neural networks that do the task that we want, but also we can ask this question in terms of biology, evolution, and it is important for our brain, the firing rate of the neurons, or the specific spiking time that each spikes happen? So this is a question, a philosophical question maybe in the field. But, and the answer, which is not clear and perfect in the community, but could be it's different depending on which task you are looking. For example, for muscles, it's typically important firing rate. If the neuron is firing more or less, the muscle will be rigid and you are going to move. So in specific situations, there is there are few difference if the neuron spikes now or 1 millisecond after or before. Despite of that, in other experiments, and for example, in some plasticity rules, it is very important the precise time difference between the spikes of two neurons. So if you do a simulation and the firing rate is the same, but the spiking time difference is different, is changing, so the plasticity rule will change and the phenomenon can be different. So for me, it's both. Like, both are important and dependent on the evolution, and the systems goes for different answers for different tasks. But it still is a question. So not only for decide which is better for your model, but also to the biological importance of firing rate neurons and spiking neurons. Questions? So, okay, more or less this is what I would like to say. First part, choose the neuronal model, check. Now we could go to synaptic models. So, questions about that? A moment for happiness, just, okay, we can think of that again. Okay. Can we build a machine that can do a specific task? Or how the brain does a specific task? So please do this specific task for me. Can you read that? Yes, it's nice, no? Or no, you cannot read, no problem. This is a problem in natural language process. So machine learning era is how to complete, and you can build different neuronal networks to solve these, and we have all these ChatGPT things. But I mean, it's something that our brain can do, and it's interesting, and we can also use biological networks to try to say what's going on when we are completing the sentence or when we are reading sentences that are normal. So this could be a research question. Let's go to a neuroimage machine. Let's measure the activity of our brain and compare when we are reading tests like that and normal tests. There are a lot of neurocognitive groups that do things with reading, and they can compare if you are reading words that have a meaning, or if you are reading Jabberwock, which are words that has no meaning. It looks like a word, the structure looks like a word, but it's not. There is no meaning. And then what's the difference with your brain signal? So then, which is a biological neural network that can reproduce this kind of activity of your brain when you show words from pseudo words. Or when you show a list of words that are not connected, that are not correlated, or when you read a text that they are correlated with each other. So this is a possible kind of question in neuroscience. And a kind of question in machine learning, in the sense that can we build machine learning that understand this text, or how our brain understands this text. So this is a kind of task that is useful for both questions and both. So This is another one. Tell me the color of each symbol. So think of the color or whatever. I think it's pretty fast. Oh, like blue, green, red, yellow, orange. I don't know. And if I do now, can you do that? Yes, you can do, but it's harder. No? So probably if you train a machine learning, there is no much difference for the machine learning. Recognize that this first word is blue and this circle is blue. But for us, it's hard. Because perhaps the binding problem that we mentioned in the beginning, because we are activating, even when we don't want, we are activating that is blue and it's circular. Okay, but they are different features, different things. But then when we do that, oh my God, it's red, blue, blue, green, yellow, red. Nice, no? I mean, at least you have funny things to talk to your family when you do this kind of research. But of course, you can really try to address in a scientific way using these ideas. So if you want to train your machine learning, your artificial neural network to do the correct answer, probably doesn't matter if it's word or if it's geometrical shapes. But if you have a biological neural network, it will be probably very different, and you will need to do a lot of choices about the dynamics of the neurons, and also of the synapses and topology that we are going to see. Questions? No questions? Okay, tell me if it's a dog, same thing. We can consider that as a task of the classify, but that you cannot see the effect of the colors. So now we can go to the synaptic model here. So how can we connect the two neurons? So we know that we can do a simulation now of one neuron here. We can do a simulation of another neuron. We have equations that describe the activity of one neuron, the activity of the other. Okay, what can we do? We can connect in the simplest way, the simplest network, biological neuronal network. But what is the synapse? So biologically speaking, it is complicated. There are many kind of neurotransmitters that can be released and combined to the synapse happen. But for us, it will enter as a current here. So each presynaptic neuron send a current to this postsynaptic neuron will make the equation of the second neuron have a new term, a new factor here coming from the synapses. Does it make sense? So biologically, the synapse will send more current to the neuron, and we want to describe how this current will arrive here, if there is a function, if it's just constant, or whatever. In a while, we'll see that we can use that, but if we go for more biological things, typically there are two main types of synapses. So the electrical synapses that are made of gap junctions that are more like a circuit, a direct connection here. It's easy. It's more or less what we, as a physicist, think when we think about synapses. So there will be a flux, an internal flux. But the more typical one are the chemical synapses, and they have a dynamical behavior, and they depends if they are ionotropic or metabotropic in time, and we're going to see a lot of these details. But when we do much of simulations of cortex and thalamus and big systems, these chemical synapses are more important. In specific cases, we use these electrical synapses here. So in these electrical synapses, the two neurons are linked together. They are very fast. They are bidirectional. I mean, they depend on the potential difference of one neuron and the other. So depending on the potential, the difference potential will enter from this one to this one or the opposite. So this is not common at all for really mainly cortical things, but they are sometimes similar to simplified networks that we try to use bidirectional connections. So excitation inhibition can happen in the same synapses, so we do not define the neuron as an excitatorial inhibitor, which is also not common in the other things. So mainly the exception is to model the electrical synapses, but the intuitive thing when we think as a physicist is the first thing that we think on how to simulate the connections of the neuron. So if you have one neuron and the other neuron described by the electric circuit, if you put just an extra connection here with a specific conductance, then you have a gap junction. So if the presynaptic neuron is doing a actual potential here, you're going to pass very proportional, a current that's very proportional to the input. Okay, I think that's all about electrical synapses. But if you want to simulate, you have to use the current here that the second neuron can receive. But in this case, the first and the second will receive this current here. So now, if we are going to chemical synapses, so we now can say about excitatory or inhibitory synapses. They are not trivial in the sense that excitatory synapses, they typically facilitate the other neuron to spike, but not necessarily. And inhibitory synapses typically inhibit, but not necessarily. So this is a common mistake. I mean, when you arrive to the field, like, okay, excitatory is just a positive current and inhibitory the negative current. But no, dynamically, they are very rich, and you can have many different behaviors from excitatory and inhibitory. But the neurons, they have this identity. So you start to mix what is a excitatory neuron is a neuron that just sends excitatory synapses, and an inhibitory neuron is a neuron that just sends inhibitory synapses, chemical synapses at least. But then the neuron is characterized by the type of synapses he does. And this is a very important difference. So this makes difference. And I think new models of artificial neural networks start to think about inhibition and how to implement that, but typically with this idea that the inhibition would be just make the other neuron to decrease the activity. So, and then since the chemical synapses now have dynamical activity, it's not just a flux of constant current. We can see if this dynamics is faster or slower. It depends on a lot of difficult names for people who are not used to. I will talk about few of them, but at least these AMPA, GABA, and then MDA, it appears a lot, and we are going to have a model for each type of these synapses. I think in the other. And just before that, the synapses produce a postsynaptic potential. So I've shown in the very beginning in the actual potential that we can arrive here and here the synapse, imagine like that. This is V and T, and if we are before the threshold, we are here, and then arrive external stimulus, it increase and start to decrease until another one arrive, and then increase, and we start to decrease, another one arrive. Eventually, it reach the threshold, and there is a spike here. So this behavior, this is a post-synaptic potential. So this is how much the activity of the neuron, the potential of the neuron increase when a synapse, external synapse arrive. So if the synapse is excitatory, we call that a EPSP, excitatory postsynaptic potential. If the synapse is inhibitory, it happens at IPSP, inhibitory postsynaptic potential, and they are represented here. So when the presynaptic neuron is spike, if the presynaptic is excitatory, there is this increase, which is this increase here of the postsynaptic neuron. And if it's the presynaptic is inhibitory, there is this decrease, which can be larger or not. So there is this inhibitory postsynaptic potential. This is also important for our models. So especially because many kind of electrophysiological measures, like local field potential, when you stick a needle inside the brain, you are measuring more postsynaptic potential than really currents. So we can model, we can use equations for these synapses. Here we have similar to the current for each ion channel in the Hodgkin-Huxley model for sodium and potassium. Let me see if I have. I don't have. Okay. This is just the current is equal to the conductance times the potential. So the conductance G now can change in time, or the conductance G can depend on the number of open channels or not. So this is very similar to what appeared for the sodium and potassium. But for the sodium and potassium, it appears M to 3, H, N to 4, okay? But it's very similar. So the synaptic current here, depending on the neurotransmitters, AMPA and NMDA, GABA A and GABA B, will have different behaviors and will be modeled in different ways. But main of this behavior just the current increase and decrease. So we can just say that we have G changing time, and we can look for these functions here. One typical model of synapses to say that this G is a function of time and this R is fixed. So if this G is like this exponential, we have this behavior. If it's an alpha function, which is very common in models of large populations, you can have this behavior here, but they are very similar. And if you have these more complicated two exponential, they are still similar, and they are more or less similar to the biological ones that measure. So when you are not really saying if the synapse is an AMPA or GABA or an NMDA, you just say that all the synapses are described by one of these functions here. Okay, so you don't, in this case, you are not adding an extra differential equation for your model. You are just adding a new term, okay? So the postsynaptic neuron is CdV/dt of summation of all currents that you are interested to, and then the summation of all synaptic currents that arrive. Here we have G and A, M. Okay? Okay, so this term, when you model with alpha function or exponential function, will be just I equal G of t and G it's the conductance exponential of t minus tj tau. So when the signal arrives, it increases, and then it starts to decrease exponentially or whatever. So you are just adding this term here, okay? But you still remain with just one differential equation for the model. Another option is now model the synapses as we modeled the ionic channels with this first order kinetic energy. Here is N and here is R, but it's R, okay? So if R are the number of ionic channels related to these synapses, so now we have a fixed conductance in time, but R the number of channels that are open or closed, they change as a first order dynamical equation. So if these channels are open, there is a chance that they close in the next step with beta. So the proportion that are closed, 1 minus all this n, has a chance of alpha to open with alpha here. So the variation of the number of channels that are open will be proportional to alpha, the numbers that are closed, minus beta, the numbers that are open. Oh, okay. Great, thank you. The receptor recycle? I don't know if I know what is the recycling? I have no idea. No, sorry, perhaps I should know, but I mean, they measure for AMPA and MDA, GABA and GABA-B, and they verified that the fast ones that are AMPA and GABA-A, it's sufficient for first order dynamics, and the others need second order dynamics. So something happened, then something happened, and another one closed, but I don't know if they are related. I mean, the biological mechanism that is responsible for this first order in this case, or the second and third order in the other case, I don't know. Okay, so typically we do all excitatory synapses AMPA and inhibitory synapses GABA-A, but when we are interested in learning, in plasticity, perhaps it's really necessary to add NMDA synapses, and the plasticity rule can be applied in AMPA and GABA, but in biological mechanisms, typically it's more common in NMDA. So we have to be careful to the way that we're gonna do this choice. So now we have the recip. Okay, we know how to model synapses, so we can choose just say that the synapse is the easiest way could be just it's a constant current, but typically the simplest one is this exponential. So to reproduce the EPSP. So just this G is our double, okay? It's our weight. All the time I'm talking about conductance, G, but in artificial neural networks, we are talking about weights, but are the synaptic weights. And what I'm trying to say is that in these biological neural networks, it's not just the weight of the synapses that matter. It's the dynamics of the synapses, okay? So sometimes we really say that the weights are not changing, we don't have rules for changing plasticity, but still when the synapses activate, there is a flux of current that goes to this neuron, and not just y times x. So there is this time evolution that is important for us and that make the difference in the moment that the synapses arrive. And this is hard to do the parallel with the artificial neural network when you are just training to classify. You don't have time. You are not looking for that, at least. So apart from this difference, what we could do, this very simple perceptron, just 3 neurons, we could do simulation of that. Okay, one question would be, can these neurons do AND or XOR? But typically the question is not that. So when we do simulations of few neurons, we now are in the moment that we can do that. We have models for neuron, we have models for synapses, but typically it's not as a classifier. We don't slice the brain, do a culture, go to the dish pattern, measure and show something, and do a stimulus and see if it classifies the stimulus. It's not the kind of question we do. We typically see what is the dynamical activity that emerge from our network. Okay, you could think that if you look, oh, I do a stimulus this way, and the answer is that way, and if I do a stimulus in a different way, if the answer is different, this can work as a classifier for us. But what we typically do is that, okay, now we are able to simulate neurons, and we can ask simple things as, do these neurons synchronize? So if we have 2 neurons and 1 synapse, then we simulate it. We have 1 neuron here doing this activity in time. And then we have a second neuron that if the synapses do not exist, would have an activity that not necessarily is correlated to that, depending on the external current that I'm putting in both of them. But then, if I put a excitatory synapse described by all these, now I have dv dt for this neuron and dv dt for this neuron, which could be the same function if they are the same model, but this one plus we receive the synaptic current here. So if the synaptic current is described by this open channels idea, we will have one more equation. We can have, if it's a Hodgkin-Huxley neuron, we would have 4 here, but then now we have 1 more for the synaptic channels that are open. So in this, we will enter here. And then we can do the simulation and see if these neurons synchronized, go to a phase locking regime, and measure the time difference between these 2 neurons. So then we can do an experiment. We can put 2 neurons together, or we can do a slice and look for neurons that connected naturally with each other, and we can measure these 2 neurons and see in which conditions the activity of these neurons synchronize and go to a phase lock. So we can pharmacological do some kind of change here to increase or decrease the weight of the synapse, and we see what changes in the dynamics of the experiment. Then we change in our model and see if we can change the dynamics here, okay? So this is the kind of question that we can answer with the very simple biological neural network that would look like as a perceptron, but in a different way. In a biological point of view, we're not thinking about machine learning, but just thinking about what a biological neural network can do in a biological system, in a tissue of brain, a slice of brain. Can we understand better the neurons if we use a biological neural network model that is very simple to compare to experiments? So then we can start to find phenomena that could be interesting for statistical physics, but not necessarily for artificial intelligence. Oh. Yeah, yes, like bidirectional. Sure, and if it's chemical synapses, we will have one equation for this, so these will have this one, and the second one will have another one. And they can be different. One can be higher, like a larger weight, or if this neuron is excitatory and this neuron is inhibitory, so this synapse is different. They can be both excitatory, but one's AMPA and the other NMDA. So there are, and you can see if they synchronize. So there is a lot of dynamical systems problem that can be reduced to this kind of network. So it's a problem of complex systems, so complex networks in a very simple way, you know, but you can study synchronization, oscillation, and things that physics are more interested in, not necessarily biologists and neuroscience. But if you can compare, like to do pharmacological changes here, then this could be useful to understand what's going on in biology, what is important for a neuron to fire more, and this could be useful if the neuron starts to lose the myelin, and this is important for a lot of neurological disease, so it's important to understand in the model what's going on and to test in experiments. More questions? I really like circuits like that. I start my life in neuroscience like that because these time differences can be non-trivial if you add very simple ingredients, like if you add a loop here, and this is possible to happen. So autopsies are common in biology. Inhibitory autopsies is somehow the neuron touch its own self in another way, and it can send a current to itself. And the dynamics of the system start to change. And if you describe the system as differential equations, these would be described by delayed differential equations, so which is mathematically richer. And then you can have new kind of phenomena. For example, cows, even if you are not with 3D equations. And these also inspired artificial neural networks to have these internal loops, but I mean, I don't know how mathematically it could help in an artificial neural network, but it's something that is biological meaningful. At least it exists in the brain. It's work like a feedback, and this kind of self-feedback could be useful. I like it. Questions? I think I can stay here, or oh yeah. Desculpa.
I know that we have some technical difficulties in terms of the physics of it and the molecular biology, but in terms of the computational process capacity, what is our perspective in the area for us to be able to simulate on a larger scale those neurons and to surpass this level of precision?
Very good question. The state of the art is trying to, like these very big brain initiatives around the world, they are trying to do the most biological details, 1 cubic millimeter of cortex of a rat. So, but I don't know even, can we estimate, like in 10 years, I don't know, the growth rate is unpredictable, I think. So 10 years ago, people are thinking, I don't know, people are being, it's recorded. They have very big plans in these initiatives, so they think that in 10 years they would understand the brain, or they told that. No, so still we don't understand. And we cannot simulate much more than 1 cubic millimeter. But philosophically speaking, the C. elegans has 300 neurons. We know all the wired thing, like how they are connected. And still, we cannot reproduce a C. elegans. I mean, like just doing a biological neural networks, and it's not alive. So there are many ways. I mean, we are trying to reproduce the brain, 1 millicortical of the brain, but when we reproduce 300 neurons, we don't know what to do with that. I mean, we cannot, I don't know, understand everything. If, just the beginning of the question, if I want to do that, or? Both, yes. Patterns typically are easy. But behavior, no. Nice question. That's it. Please. So tomorrow we talk about learning, memory, and synaptic plasticity, then topology, and then other applications. But I hope you can internalize like a lot of information. Oh, a dinner. Nice.